Hello, my dear students. I hope you all have been doing well. Um, from today, we are going to start our new topic that is about protein metabolism. And in this topic, first we would be studying about the digestion and absorption of protein. In this lecture, that is lecture number 10. And after this lecture, in lecture number 11, we would be studying about catabolism of protein. And in lecture number 12, we would be studying about the anabolism of protein, that is the amino acid. And then after that, in the next week probably, we would be studying about the catabolism and anabolism of heme and the porphyrin compound. So this would be a whole topic that would complete in 2 weeks, that would be about the protein metabolism. So in this topic we would be encountering with many different pathways of protein and amino acids, biosynthesis and catabolism. And we would be seeing that how the digestion, protein digests in our body and then absorb. So starting from here, this is my lecture number 10, and in this first we are seeing that how protein digests in our body, and then after digestion it absorb in our body. So as before starting this lecture, before starting to understand about the how protein digests in our body and how protein after digestion absorb in our body, we first have to know about what are proteins. So what are their basic structure and how it is made up of. So here we are seeing that protein are, what are protein? Actually protein are macromolecule like other macromolecule of our body, means like uh, carbohydrate, lipid. Protein are also macromolecules. So um, why we are seeing that protein are macromolecule? Because they are made up of amino acids that are monomer. So here in this picture we can see that we are having a polypeptide chain. So this polypeptide chain, it is a chain of amino acid. So protein, what are protein? Protein are larger macromolecule that are made up of um, um, amino acid monomer. So what are the amino acid? Amino acid or amino acid are the organ compound. These are the nitrogen containing compound that are having one amino group, one carboxylic group, and one the R group. So we know about this, that in nature we are having 21 amino acid, 20 or 21 amino acid, and all these amino acid, this is the standard rule that all the amino acid would be having a amino group, a carboxylic group, or a nitrogen atom, and the one of the R group. And in each amino acid, this R group would be changed. Means each amino acid would be having this different R group, and because of the difference in this R group, we are having different amino acids. Some are polar, some are non-polar, some are means hydrophobic, some are hydrophilic. So this was all about the amino acid. What then amino acid to do when these amino acids join with each other via a bond that we say as peptide bond. So after joining this amino acid via peptide bond, we will be getting a long chain that we say as polypeptide. So these polypeptide chain, many different polypeptide chain means collect with each other and form means secondary and tertiary structure and then quaternary structure of protein. And how this polypeptide, how this amino acid join, these amino acid join with each other, how this amino acid join and how they form a polypeptide chain. So the amino acid join with each other. We can see here in this reaction what happened, that hydroxyl, we are having two amino acid. and one is having R1, one is having R2. So it means we can understand that one is these amino acids are two different amino acids, or they are going to join via polypeptide chain. So how this chain bond would be, uh, these are going to bind with the polypeptide bond. So this how this bond would be formed. The one molecule of hydroxyl, one hydroxyl group would remove from this molecule, and one hydrogen would remove from this molecule, and it will come out from this both these molecules as a water, and a bond will be formed between a between this group and the nitrogen of this between the carbon and the nitrogen of two different amino acids, and we say this bond as a peptide bond that join two amino acids together, two amino acid residue together. What are amino acid and what are amino acid residue? Amino acid is a molecule that are having a carbon atom that is attached with one carboxylate group, one amino group, one nitrogen, and one R group. However, amino acid residue is a molecule, or we can say it is a nitrogenous molecule that is having, or it is a amino acid means that is having uh, that is not having means that we made that we get after removal of water molecule. So what happened there? This is basic of the protein. What are protein? Protein are, we can say, that collection of monomer that are amino acid, and protein are the polymer of amino acid, you can say in other words. So you have to keep in your mind that protein are the macromolecules that are made up of uh, amino acid. So if we will be talking about the digestion of amino acid, first I am talking about the digestion of amino acid. So what does mean by digestion? Digestion means whatever protein molecule you are taking in your mouth from outside of your body, or whatever the protein molecule that are present inside your body in this form. This is a protein polypeptide chain. So in this form, so what will be the digestion? Digestion will be the breaking down or chopping down this long polypeptide chain into smaller pieces, or even the breaking down of this peptide bond, so these two amino acids can be separated from each other. So after the digestion, what we get means digestion of protein start from acting of enzyme on the large macromolecule of protein, and then after that what we get at the end, we get simple this amino acid. We simple separated amino acid at the end. So here we are seeing this two molecule of protein, two molecule of amino acid joining with each other through a polypeptide bond with the removal of a water, and forming and start forming a polypeptide chain. So this is the anabolism, we can say. And what will be the catabolism? When this reaction would get reversed, means this the two amino acid, or these short two you can say short we can say it diapeptide. We can say that what would be the catabolism of protein when the peptide bond between these two amino acids would be break, and it will release these two amino acids. So it will be the catabolism. So what happened when uh, uh, um, uh, uh, the digestion take place? In the digestion, what happened that the enzyme break down this polypeptide, this peptide bond between the amino acid. So if the enzyme uh, will break these peptide bond from here, so what we will be having? We will be having diapeptide. Means if enzyme would break this polypeptide chain from here, so we will be having three amino acids that will be removed from this polypeptide, and this three amino acid 1, 2, and 3 would be joining with each other through two peptide bond. So we will be having at the end a diapeptide. Okay, and then if the enzyme uh, would means break the peptide bond from here. So what we having in the end, we will be having triapeptide. What does it mean? That four amino acid would be released from this polypeptide, and these four amino acid would be joined with three peptide bond. So in this way, the enzyme work on this our body enzyme we will be discussing in our coming slides. So by this way, our body enzyme would act on these polypeptide and then convert this large polypeptide into dipeptide tripeptide. So in the next slide, we are going to see, we are here, this you can say this is the summary of all the means enzyme that take part in this process, or all the mechanical support, or all the summary how the mechanical and the chemical digestion takes place in our body. So as all other macromolecule that we have been discussed right now, till now, means means carbohydrate, lipids. So as we saw in in that case, the for the digestion and for the digestion of for example other macromolecule, carbohydrate, lipid, both mechanical and chemical digestion help for the for the digestion of carbohydrate and lipid. So similarly here, if we would be so similarly here, if we are talking about um, the protein. So protein digestion, I'm sorry, there was some problem. So if we are talking about protein, for the protein, the same thing happens here. Means the protein digestion would takes place by both mechanical and the chemical support. And what are these mechanical and chemical support for the digestion of protein? And from where it comes in our body. So this mechanical and chemical support is the chemical support is different enzymes that are present in different part of our body, and the mechanical support is the means the muscular contraction of the different part of our body. So in this diagram, in this slide, we would be studying that how protein means the digest with the chemical and mechanical support. So first of all, we have to understand that protein digestion takes place in our mouth, in stomach, and in intestine. These are the three main organs where protein digestion takes place. And if we talk about the digestion of protein in the mouth, so in mouth, saliva helps for this digestive process, and mouth means chewing activity of our teeth, and the muscular contraction of our jaw. So all the buccal cavity, all these help for the mechanical digestion of protein. And in stomach, different gastric means secretion help for the digestion. These include the hydrochloric acid and the enzyme that is pepsin. So in next slide, we would be studying about what this pepsin means does, how this pepsin take part in the digestion of protein. First of all, you have to just keep in your mind that in the stomach, for the chemical digestion, hydrochloric acid and the enzyme pepsin help for the chemical digestion of protein in our stomach. And for the mechanical digestion of protein, stomach churning movement help for the digestion. And in the intestine, intestine peristalsis movement help for the mechanical digestion. And then for the chemical digestion, pancreatic enzyme that are trypsin and the chymotrypsin and the trypsin. These two enzymes help for the chemical digestion. And in the intestine, some other enzymes are also released in the intestine from the intestine itself. So these enzymes also help for the digestion of protein in the intestine before absorption. So this pancreatic enzyme, from where they are coming from, they are releasing from pancreas and coming into the intestine, where they are helping for the digestion of protein. So in the next slide, we will be seeing that how this protein would be going through the mechanism of digestion from your mouth to the intestine. So you have just to keep in your mind that digestion takes place. Digestion of protein starts from the mouth and ends on the small intestine. However, the absorption takes place in the intestine. So what happens if you are taking food in your mouth? So if we would talking about the protein, so here we are taking an example of protein, that is eggs. Egg is full of protein. So what type of protein is present in the eggs? In eggs, albumin is present. That is one type of a protein, egg protein. So we are talking about the digestion of the albumin, that is the egg protein. So when you take this egg protein in your mouth, so what happens? You will cut this egg protein or egg into small pieces by the help of your teeth. And then this is the mechanical digestion. And the salivary gland release the saliva that means make this help in the digestion. How? Because it just wet this content. So then you will means broken down the protein into smaller pieces, and after that in the form of a bolus, this means partially digested protein would be engulfed and reaches to the stomach. In the stomach, what happened? The stomach gastric juices having the hydrochloric acid and the enzyme pepsin that help in the digestion of protein in the stomach, that help for the chemical digestion of protein in the stomach. And the stomach churning movement would help for the mechanical digestion. So what does this pepsin do? Pepsin is a proteolytic enzyme. What are the proteolytic enzymes? Proteolytic enzymes are those enzymes that hydrolyze the peptide bond, or that hydrolyze protein, or that hydrolyze peptide bond. So what happened? This pepsin, what does it do? It break peptide bond by adding the molecule of a water in that. So what it do, if you are having a long polypeptide chain that reaches to the stomach from your mouth, so you are already having an idea. If we will go from the to the next the previous slide, if you are having this long polypeptide chain in your stomach, so what this pepsin would do, this pepsin will act between these amino acids and then add a molecule of a water in this peptide bond. So what you will be having, you will be having this triapeptide separated, and all this polypeptide chain will be also present in your stomach until and unless it would get chopped into diapeptide or tripeptide by this pepsin. So pepsin can will break this will break the peptide bond uh, among different position of this peptide and chop into smaller pieces of diapeptide and tripeptide. So this and the what this hydrochloric acid will do, hydrochloric acid will help this enzyme to perform its catalysis, because some enzymes are work on the acidic pH. So what happened then? This dipeptide and tripeptide then will reaches into the small intestine, and in the small intestine, what happened? Small intestine, this dipeptide and tripeptide would then further break down. Means if you are having dipeptide, so what does it mean? It means you are having a chain, a small chain. If you are having dipeptide, what does it means? If As I have previously discussed with you, if you are having dipeptide, what does it mean? You are having a small chain like this. Here we can see a chain of means three amino acid in which you are having two peptides. So you have to separate all the amino acid from this small fragment that is having three amino acid and two peptide bonds. So what dipeptide is, what the enzyme in the small intestine do, these small intestine would again break the remaining bonds that are present in the between the within these dipeptide and tripeptide. So in the small intestine, you are having the enzyme that is dipeptidase and tripeptidase. So this dipeptidase enzyme only work on the small chain that is having two peptide bond and chop it into this and release the separate amino acid. And tripeptidase would means act on the small chain of small polypeptide chain that is having three peptide bond. So after the breaking of these three peptide bond, what we will be having? We will be having small means separated amino acid or individual amino acid. And then what happen after this? When you will be having separate amino acid, these amino acid would be transport from your intestinal lumen into the microvilli cell by the with the help of transport protein. So after the entrance of these separated amino acid in the microvillus cell, these are transported into your blood vessel, and from the blood vessel they would be transported into your liver, and then liver will be having all these amino acid in your liver. And these amino acid, what do means liver would decide that from which part of the body these amino acid should be transported. So first of all, you have to we will be discussing more about the liver in the coming slide, but here you have to keep in your mind that this was the whole digestion of the protein. Means protein digested in your mouth with the mechanical digestion, and then it reaches in the stomach where both chemical and mechanical digestion takes place. For the mechanical digestion, stomach churning movement helps, and for the chemical digestion, pepsin enzyme and hydrochloric help to break down long polypeptide into dipeptide and the tripeptide, and these dipeptide and tripeptide would reaches to your small intestine, and in small intestine you are having the enzyme that is dipeptidase and tripeptidase. This dipeptidase and tripeptidase enzyme would be convert these means little amino acid chain into the separated amino acid. And then what happened? Then you are also having in your small intestine the enzyme chymotrypsin and trypsin. These are the pancreatic enzyme that synthesize in the liver and reaches that synthesize in the pancreas and reaches to the intestine from via the pancreatic duct. So these chymotrypsin and trypsin, what does they do? They also break down the peptide bond. So they help to break down the peptide bond or hydrolyze the protein. So with the help of all these pancreatic enzyme and the small intestine enzyme, what happened? The these enzyme, that is the intestinal enzyme peptidase, that is dipeptidase and tripeptidase, and the pancreatic enzyme, that is trypsin and chymotrypsin, would means release small amino acid or separated amino acid from this big polypeptide chain. And then this was whole about whole digestion, but how absorption takes place? Absorption takes place by the help of transport protein. uh, on the For that we use the ATP. So by the process of active transport, these small transport protein would means transfer this amino acid into the microvilli cell. So from the microvilli cell, they would be enter into the blood vessel and then transported to the liver. From liver, they would be transported to rest of our body. So when your amino acid, when the polypeptide chain broken down into smaller amino acids, and then amino acid are reaches to the liver. So what happened in the liver? Liver decided that what will be the fat of this amino acid. Actually, liver would be having um, uh, these small stores of amino acid, that we say as amino acid pool. So this is the small means storage of the amino acid. And what does mean by amino acid pool? It means that the amino acid that are present in stored in the liver in a small quantity, that can never replenish, means they keep turnover, means they from this pool, these amino acids would be continuously using to synthesize some other protein in your body, or they may be break down and again stored in this pool. So and then what happened in from this amino acid pool? Some amino acids can be used to produce energy, some amino acids can be used to synthesize glucose or fatty acid, and some amino acids can be used for the synthesis of non-protein molecule in your body. So non-protein molecule are those, what type of non-protein molecule can be synthesized from the amino acid? Means non-protein molecule are those molecules that are having nitrogen uh, in it. We will be studying about our coming lecture what can be these non-protein molecules. It can be a urea, means when amino acids catabolized, it completely it convert into the urea that uh, uh, is split out from your body. So this was the amino acid pool that would remain uh, in your liver, and then would be used to synthesize other protein, either non-protein molecule, either glucose or fatty acid, or to produce energy. So I hope you understand about this lecture. So how protein digests? First, protein breakdown in your mouth, and then protein digestion takes place in your stomach by the action of enzyme trypsin. Trypsin is a enzyme that hydrolyzes peptide bond by addition of a water between these bonds, and then these small peptides will reach in your stomach. These diapeptide or small peptide, you can say diapeptide or tripeptide, would reach in your intestine, and where the intestinal enzyme, that is that is pepsin, and then pancreatic enzyme trypsin and chymotrypsin would hydrolyze further these protein into individual amino acids, and these amino acids then absorbed from your small intestine to the microvillus cell, and from the microvillus cell they would be means enter in your blood vessels, and from the blood vessel that would be transported to your liver, and in liver they will be stored there for a shorter period of time, and then from this store these amino acids can be used to synthesize other protein, can be used or transported. Means in liver there would not be any synthesis of protein would be takes place, but the liver transport the protein on those areas where the protein need to be synthesized. So the protein would synthesize on the other means from these amino acid, or whenever the amino acid protein breakdown in any part of your body, the means individual amino acid would reaches again to the liver, and there they would be stored for certain period of time until and unless they would be needed to means perform their work. And what would be their work of amino acid? Either they can be used for synthesis of protein in other part of the body, or either they can be used for the production of energy, or either they can be used for the production of glucose or fatty acid, or either they can be used for the production of non-protein molecule that would be containing nitrogen bond. So this was the whole lecture. These are here some links. You can go through these links and further study, or either you can consult with these books. I hope you understand about the protein, how protein digests and absorb in your body. And in our coming lecture, we will be studying about how these amino acids, simpler amino acids that we get after the digestion, uh, how these amino acids then further catabolize into um, metabolic product. So thank you very much, and Allah Hafiz.